Welcome to the presentation of the doctoral thesis of Orathai Kamsoi. The thesis is entitled Factors Regulating Metamorphosis in Metabolan Insects. And first of all, I am going to introduce the members of the committee. Here's José Luis Maestro from the Institute of Evolutionary Biology as secretary. Jan Sardà from the IRTA, but now is in the Autonomous University, right?
Yeah.
As vocal, and myself, Isabel Navarro from the University of Barcelona. As you know, the first part of the procedures is the presentation of the work by the doctoral student, and we follow it by a debate between the candidate and the members of the committee. So now, Kim, you have the floor.
Yeah. So thank you very much. And thank you everyone for coming. So good morning. I'm Orathai Kamsoi, a PhD student under the supervision of Xavier Beyes. Today I'm going to present my thesis entitled Factors Regulating Metamorphosis in Hemimetabolan Insects. So here is the outline of my talk. So I'm going to tell you in brief about the origin and the evolution of insect metamorphosis, each type of insect metamorphosis, the hormonal regulation and the molecular mechanism which rely on the MECRE93 pathway, and two species that I used in this study, the Blatella germanica and Cleon dipterum. And then I will talk about the hypothesis of the thesis and the objective. For the results, is divided into three parts. The first part is about the role of myoclonine that triggers the pre-metamorphosis in hemimetabolan insects. And the second is about the role of E93 in the destruction of the prothoracic gland. And the third part is a regulation of metamorphosis for our new model, the mayfly Cleon dipterum, and the conclusion. So let's start. When we talk about the insect, they are the largest and the most diverse insect group of animals that presently know. The first wingless insect that emerged here around 475 million years ago. And then, however, the thing that make the insect the most diverse group is not this group. Is because the evolutionary innovation of the wings and metamorphosis. So the Pterygota, or the winged insect, or the direct metamorphosis, emerged around 400 million years ago, split into the Paleoptera and Neoptera. Later, the Neoptera split again to the Polyneoptera and Eumetabola. And finally, the Eumetabola, they split into the Paraneoptera and Endopterygota, or the Holometabola insects. Two species that I used in this study. The first one, Cleon dipterum, is in the class of Paleoptera, and the second, the Blatella germanica, is the class of Polyneoptera. So both of them are quite primitive. For the post-embryonic development of insects, it can be classified into three groups. The first one, the ametabolan metamorphosis, the insect with direct development without change any significantly in form during development. In this group, they have three main stages of development: the embryo, the nymph and the adult. So the nymph are very similar to the adult, and they just lack the functional genitalia. So they increase in size by molting until reaching the adult state, and then they continue molting. And here is the sample of the wingless insect, the bristol tail and the silverfish. Sorry, could you hear me? It's okay, no. Okay. The second type of metamorphosis is the hemimetabolan or direct metamorphosis. In this type, the juvenile stage, also called nymph, but they have the external wing patch. So they can morph directly to the adult with the functional wings and the functional genitalia. However, in this group, with the with the evolution of metamorphosis, they stop molting after reaching the adult state. So the final molt is the innovation for metamorphosis insects. And here is the sample of hemimetabolan insect, the mayflies, the cockroaches, and the kissing bugs. And the last type of metamorphosis is holometabolan metamorphosis, or the complete metamorphosis. In this group, they have one more stages during development because the larva is totally different with the adult, so they need one more instar, one more stage, the pupa, for the transition. And this group of insects, they are the most diverse among insects, as you may get familiar, like the beetles, the moths and butterflies, and the flies. For the regulation of metamorphosis, there are two main hormones that regulate metamorphosis. The ecdysone is active from 20-hydroxyecdysone from the prothoracic gland, and juvenile hormone from the Cobra alata. If I don't use the microphone, could you hear me? Okay. Yeah. And the second, the juvenile hormone from the Cobra alata. So these two hormones, in the case of ecdysone, is induced molting. In the young nymph, with the presence of juvenile hormone, the insect will molt to another immature stage. But during the last nymphal instar or last larval instar in holometabolan insect, with the absence of juvenile hormone, the insect can molt and metamorphosis, change the form into the next instar. The molecular mechanism underlying the action of these two hormones based on the macronutrient pathway. This pathway starts with the juvenile hormone, binds with its receptor, the metoprene tolerance or MET, and then this complex from the dimer with thymine, another transcription factor, to induce the target genes. So during the young instar, immature insect, juvenile hormone induce the expression of Kruper homolog 1, which in turn replaces the E93, the promoter of metamorphosis. However, during the last nymphal instar, for example, in the case of Blattola, the decrease of juvenile hormone titer is interrupt the expression of Kruper homolog 1. So in this case, it has the strong induction of E93, so they can trigger metamorphosis. The main difference between hemimetabolan and homometabolan insect is broad complex, the action of this transcription factor. In the case of hemimetabolan insect, broad complex is promoting the development of wings. But in holometabolant insect, broad complex is determine the formation of the pupa. So the first model, or the main model that I used in this study, the German cockroach or Blattula germanica. This insect is a good model for study in the laboratory because it's easily to rear or maintain in the lab. And also the cockroach are one of the primitive insects that have been on Earth more than 300 million years ago. So they can serve as the basal model to study evolution or metamorphosis, especially the transition from hemimetaboly to holometaboly. And also in our lab, we use the RNA interference experiment to test many hypotheses. And this insect is very sensitive to RNAi. And here is the 3 last nympho insta that I used, the anti-penultimate or nympho, the penultimate, and the last nympho insta or NIM6. And here you will see the change of the juvenile hormone during the penultimate and the last nympho insta. The second model is Creondipterum or the mayflies in order Ephemeroptera. This insect is one of the most primitive winged insects, emerged on Earth at the same period of the dragonfly. So the thing that make them very interesting for study is not only the primitivism, but they can molt one more time after they gain the functional wings in the subimago. And also, from here you will see the nymph live in water. So they spend the nympho life in the fresh water. And then when they ready, they just molt to first to subimago and then more again to imago. So in this characteristic, they are a good model, not only for study the metamorphosis and also the evolution, the origin of wings and adaptation. This thesis is based on 3 hypotheses. So the first one is come from the fact that the clearance of juvenile hormone is necessary to initiation metamorphosis. So we are looking for, we have searched for the candidate that should play this role to reduce or to promote the reduction of juvenile hormone production during the last nympho insta. And one gene that we found that highly expressed during the penultimate nympho insta, the insta before the last insta, the last nympho insta is myoclonine. So the first hypothesis, we think myoclonine is involved in the regulation of the decrease of juvenile hormone production at the beginning of the last nympho insta. The second hypothesis is from the final mode, the innovation of metamorphosis insects. This process is mediated by the destruction of the prothoracic gland. According to E93, it plays an important role in metamorphosis. So we thought, we hypothesized it should be involved in the degradation of the prothoracic gland after they enter the adult stage or during the transition from the last nympho insta to the adult stage, also in Blattula germanica. And the third hypothesis is from the MACRE93 pathway, since we study this pathway in many Neopteran insects. And we found that this pathway is conserved in the context of regulation of metamorphosis in those species. But nothing is known in the case of Ephemeroptera or the Paleopteran insects. So we want to explore this possibility if the MACRE93 pathway operate the metamorphosis in early branching Polypteran insect. So the objective is derived directly from those hypotheses. First, to study the possible role of myoclonine in regulation of the decrease of juvenile hormone production, which occur at the beginning of the last nympho instar in Blattula germanica. Second, to study the possible role of E93 in the destruction of the prothoracic gland after the imaginal mode, also in Blattula germanica. And the last objective, to study the mechanism that regulate metamorphosis in the mayfly, Cleondipterum, particularly in the formation of subimago, to compare this mechanism with those operating in Neopteran insects. The first sections of the result, the role of myoclonine, the results summarized and have been published in FASEP journal last year. So I'm going to tell you in brief about the findings from this study. So the first question that we have when we start this project is the mechanism that caused the reduction of juvenile hormone production at the beginning of the last nympho instar in Blattula germanica. And from our transcriptomic data, we found that myoclonine, which is the ligand of the transforming growth factor family in activin branch, is highly expressed during the penultimate nympho instar. So that's why we thought this gene should play some role in this context. In addition, in the cricket, they also show the role of myoclonine in suppressing the expression of juvenile hormone acid methyltransferase in the corpora allata, and it triggers a decrease of juvenile hormone production in that insect. And the depletion of myoclonine in this species is inhibit metamorphosis. So this is our antecedents for this study. So in Blattula germanica, we first studied the distribution of myoclonine in various tissue, and we found the high expression of myoclonine in the prothoracic gland, also in brain, in corpora allata, and in muscle. But as we interested in the mechanism that control, that regulate metamorphosis, so we study the expression pattern of myoclonine in corpora allata and prothoracic gland, the two tissues that produce juvenile hormone and ecdysone. In the case of corpora allata, we found the high expression of myoclonine since the beginning of the penultimate instar. But in the prothoracic gland, there is a peak of myoclonine in the middle of penultimate instar, and the expression is kept in high level until the first day of 6th nympho instar. So with that profile, we start to do the RNA interference. We inject double-strand RNA against myoclonine since the first day of penultimate instar. Myoclonine depletion in Blattula germanica trigger the formation of supernumerary nymph. So the insect more to another nympho instar, and this is this result is the same that they found in cricket. The molecular analysis that we study, we found that when we suppress myoclonine, the expression of juvenile hormone as its methyltransferase in the corpora allata is upregulated, which caused the increasing of grouper homolog 1 in last nympho insta and the downregulate of E93 also in the last nympho insta. So this is the explanation why the insect more to supernumerary nymph after last nympho insta. So the role of myoclonine in corpora allata should be the same as in cricket, another hemimetabolant insect, in suppress the expression of juvenile hormone as its methyltransferase in penultimate insta in the case of Blattella germanica. So this produced the decline of juvenile hormone production at the beginning of the last nympho insta, which is the prerequisite for metamorphosis. In this experiment, we also study the effect of myoclonine depletion in the prothoracic gland. And this is the results that we found. We found that the prothoracic gland of myoclonine depleted nymph was bigger than those control nymph. Then with edu labeling, we found that the prothoracic gland, the cell of the prothoracic gland in myoclonine depleted, they have much more active cell division, more than control. And at the molecular level, we measure the expression of DACAPO, the inhibitor of cyclin kinase, and we found the downregulation of this gene. So it should be the explanation why the prothoracic gland undergoes the cell hyperproliferation when we suppress myoclonine. Then we measure the expression of the genes in ecdysteroidogenic pathway, the genes that produce ecdysone, and we found the decrease of the expression of those genes, like Neverland, Phantom, and disembodied in myoclonine depleted animal. So from this result, it seems myoclonine indirectly promote the expression of those genes to induce the arrest of cell proliferation in the prothoracic gland, and maybe via the transforming growth factor beta signaling to stimulate the expression of those genes for producing the last pouch of ecdysteroid, which needed for metamorphosis. And also in this study, we also found that since the nymph more to supernumerary nymph after 6 nympho insta, so it means the prothoracic gland did not degenerated after last insta. So when we measure the expression of grouper homolog 1 and E93 in the PG, we found the upregulation of grouper homolog 1 and downregulation of E93. And on the last day of 6 nympho insta, there is a decrease of IAP1, a decrease of Fujitarasu transcription factor 1. The genes that produce that induce the programmed cell death in the prothoracic gland of Blattella germanica, and the increase of the inhibitor of apoptosis 1, IAP1. So this result is tell us about why the more to supernumerary nymph And the summary for the first experiment, we can divide the role of myoclonine into two tissues. First, in the corpora allata, the high expression of myoclonine during the penultimate lymphoid instar replaced the expression of juvenile hormone acid methyltransferase. So it results in the reduction of juvenile hormone production at the beginning of the last lymphoid instar. In the case of protocyst gland, high expression of myoclonine that happened from the mid-end of the penultimate lymphoid instar until the first day of the last lymphoid instar. It should help to replace the cell proliferation in the protocyst gland. And also, it correlated with the high expression of ecdysteroid synthesis genes for promoting the metamorphic ecdysone pulse which needed for metamorphosis. And the side result that I will explain more for the next section. We found that depletion of myoclonine prevents the onset of the PG degeneration by decrease the expression of E93 in the protocyst gland. And this cause is maybe it raises the question or the hypothesis for the next section that E93 might be involved in the degradation of the protocyst gland. So then in the second section of the results is about the role of E93. For the summary of this result now is underway for publication in the journal Development, and we just revised and resubmit last month and hope it will be accepted soon. Yeah. So the antecedents for this study, first from the previous study in Blattella germanica, it showed that the histolysis of the protocyst gland is regulated by ecdysone signaling by upregulation the Fujitarasu transcription factor on the last day of lymphoid life, as you see here. And when they deplete Fujitarasu transcription factor 1, they found that the PG did not disintegrate. Also, in the previous work of my thesis, when we deplete myoclonine, the animal moved to supernary nymph after NIM6. So, and there is the effect of the expression also for Fujitarasu transcription factor 1 and E93 in the protocyst gland. So these two genes is down-regulated. So this suggests that E93 might be involved in the destruction of the protocyst gland also. And in Drosophila, in the case of E93, they already study and demonstrate that E93 is a key player of the histolysis in many tissues like salivary gland or in the midgut and also in the fat body, which happen during metamorphosis. But in Blattella germanica, the role of E93 in the destruction of protocyst gland, we never study. So this is the antecedents for the second topic. So first, we start with the study of the expression of E93 in various tissues. First, in the protocyst gland, in the corpora allata, in epidermis, represented by the abdominal turgis, and in wing patch. And we found that the expression of E93 also Fujitarasu transcription factor 1. We measure both genes together. It's different with the other tissues that we investigate. So from this result, we start to suppress E93 by injecting double-strand RNA against this gene in the late NIM6 or in NIM6D6 with the aim that we want to suppress E93 only in the prothoracic gland, but not affect the other tissues. Because on NIM6D6, the expression of E93 in other tissue is already past the highest expression. So, but it still comes to the prothoracic gland on the last day. So the result that we got after that, first we found that the nymph can mold to the normal adult with more or less the normal shape when compared with the control, and they preserve the prothoracic gland. Then we measure the expression of E93 and Fujitarasu factor 1 in the prothoracic gland on NIM6D8 just to test the efficiency of the double strand, and also we found that when we deplete E93, the Fujitarasu transcription factor 1 expression did not go down. So it's not affect those genes. And also in NIM6D8, we also measure the expression of IAP1 and caspase1, two genes that play the opposite role in the programmed cell death. And we found the upregulation of IAP1 in NIM6D8, but for caspase1, the effector caspase, we found the downregulation of this gene on adult day 1. So these results already show, already explain the reason why the gland did not disintegrate when we suppress E93 on late NIM6. And also we also found that the prothoracic gland in this animal is still function because they have the active transcription of the genes in ecdysteroidogenic genes is fully active. As you see here, the high expression of Neverland phantom or disembody are also shadow in this particular insect. And the expression of the genes for ecdysone dependent E75A and HR3A was also expressed in the highest level when compared with the control that we cannot detect the expression of those two genes in normal adult. And the level is more or less compatible with the expression in NIM6D6, which is the day that the insect produce the pulse of ecdysone for molting. So in this study, the most striking result is not only the prothoracic gland did not degenerate when we suppress E93, but we found that those adults that molt after deplete E93, they are able to molt again. So on day 9 of adult life, we can observe the new mandibles under the old ones. And on day 10, this adult try to undertake ecdysis, but they cannot complete. Since you see here just some part of the cuticle that can shed or remove. And also in the wings, we also found that they try to do apolysis. And also they form the new veins under the old ones. And we checked for the expression of wing-related genes in wings of these adults, and we also found that the expression of those wing genes is high expressed. Moreover, we just try to explore more how is the epidermis, the type of the cuticle that this adult produce. So we measure the expression of the cuticular protein genes. The first group is the nymphocuticular protein, this one. And we found that they still express the nymphocuticular protein genes in the high level. But in the case of adult cuticular protein genes, the expression in 83 treated adult more or less is similar with the control. So from this result, we can consider the cuticle of E93 depleted adult. In general, it should be the cuticle of adult, but with some nympho character. And this may be, then this gives us a clue like why they still respond to ecdysone in the epidermis. Then we measure the expression of the ecdysone signaling genes, the first ECR and RXR, which is the receptor for ecdysone. And we found that for these two genes, the expression of those two genes in control and treated is more or less the same. But in the case of E75 and HR3A, we found the high expression of those two genes in E93 depleted epidermis. So then we think this control adult that cannot molt, maybe they don't have ecdysone. So we did another experiment. We inject 20-hydroxy-ecdysone to the control adult. But with two doses that we used, the physiological and pharmacological dose, 10 times more for the physiological one. All of them cannot upregulate the expression of E75 or HR3 as we expect. And we left some adults just to see what happened after that, if they can show some symptom of molting, but it did not happen in this adult. So from this result, we presume that the molting in E93 depleted adult is not due solely only the high level of ecdysteroid, but it should be happen because there is some factor from the prothoracic gland that can make the epidermal cell that are competent to the steroid and can produce the new cuticle. And also, we think the adult commitment of the epidermal cell of in the insect, maybe it should be involved with the E93 or Fujitarasu factor 1 should play some role in the adult commitment of the epidermis. And also, the injection of ecdysone, maybe they cannot produce the more precise pattern of the increase of ecdysteroid that may be crucial, but it's crucial to trigger the appropriate cascade of the expression of the genes. And the last experiment in this study, since we wonder how is the interaction between E93 and Fujitarasu transcription factor 1. Because when we suppress E93, we still get the adult with the protocyst gland that did not degenerated, same as when they suppress Fujitarasu factor 1 in NIM6. So we did this experiment to test that hypothesis. Inject double strand against Fujitarasu transcription factor 1 on day 7 of NIM6, 1 day before the peak in protocyst gland. And we got the adult, the normal one, after deplete Fujitarasu transcription factor 1. But when we measure the expression of E93 in the PG, we found the down regulation of E93. So if you remember the first result that I showed when I suppress E93 in the PG, when the expression of E93 is going down, the expression of Fujitarasu factor 1 did not go down. But this time, when we suppress Fujitarasu, it reduced the expression of E93. So from this result, we can conclude that the effect of Fujitarasu transcription factor 1 in the degeneration of the protocyst gland, it should be induce or activate the expression of E93. And E93 is the effector of the destruction of the protocyst gland in this animal. However, in this study, we also found that the activating role of Fujitarasu transcription factor 1 on E93 is specific just only with the protocyst gland. Because in the case of Corpora alata, we did not see the decrease of E93. Okay. And this is the summarize of the second section. So first, E93 depleted nymph can morph to adult that preserve the protocyst gland, and this gland is function, and it cause the animal can morph again in the adult stage. The second, the morphing in E93 depleted adults is not due to solely the high level of ecdysteroid, because when we did the treatment of ecdysone, the control adult did not morph, so it might be involved with some factor, maybe from the protocyst gland or the factor that make the commitment of the epidermal cell, which involved with E93 and Fujitarasu transcription factor, factor 1 in somehow. And Fujitarasu transcription factor 1 depletion at the end of the nymphal life is down regulate the expression of E93 because of but the effect of this gene on E93 is specific only to the protocyst gland, and it leads to the conclusion that the death of the protocyst gland is induced by Fujitarasu transcription factor 1 is mediated by E93. And next, I cannot stop now because I think I'm running the time, so I will continue until the last section. So the third result, the third section of the result is the regulation of metamorphosis in the mayfly, Cleondipterum. So in this section, we are preparing the manuscript for publication. We need more experiment to complete the manuscript, and we hope we will finish soon. So the antecedents of this topic is come from the study about metamorphosis in Neopteran insects. We know that the regulation of metamorphosis in this group of insects is condensed in the macro-93 pathway. But in the Paleopteran, like in the case of mayfly, very little is known about the regulation of metamorphosis. And the lack of this information has the impact that we cannot complete the picture of the evolution of metamorphosis or evolution of insects. So that's why we want to explore the control mechanism of metamorphosis in this species. In the past, we cannot do that because it's not easy to keep these insects in the laboratory. But now with the successful of the continuous rearing system of this species, Creondipterum, so it has been developed recently, and it allows us to study the metamorphosis under the experimental condition. Since we don't have since we don't have much information about these insects, so I start the study by trying to characterize the nympho stage of Creondipterum. And just as I interested in metamorphosis, so I focus on the last 4 nympho insta preceding metamorphosis. So we use the length of the wing pads to distinguish or to characterize each nympho insta. And then we also found in the case of the wing maturation in this species that occur during metamorphosis. The maturation of the wings is just they have like the big change during the last 2 days of last nympho insta before molting to subimago. So here in figure C, you can see the folded wing, the black one in the pocket of the late day 4. So this is the wing, and if you remove the pocket, you will find the wings of subimago inside. And also the change of the wing pads, we also use to differentiate the edges within the same stage of this species, or maybe it can be used in other species of Ephemeroptera also. And during metamorphosis, we also found the change in the mouthparts of these insects, since the subimago and adult do not feed anymore, so they lack the mouthparts during that state also. So the new mouthparts just produced when they molt from nymph to nymph, but in the last nympho insta, they did not produce the new mouthpart in that moment. And in the case of the central filament, we found that they do not form during the transition from the last nympho insta to the subimago. And finally, for the remodeling of the last tarsomere, we also found that there is a change of the last tarsomere from nymph to the subimago. They will change from the elongate and the claw shape in nymph to become a shorter one, and with the hooked shape in subimago and in adult. And you can see this change clearly on the last day of the last nympho instar. For the growth of the forelegs during metamorphosis, we found that the forelegs of Creondipterum can grow double in length during this transition. So then with this information of the morphological study, I will use it when I do the next experiment with metoprene. So from the result of morphological study, we found that the most stages that have the change from metamorphosis is during the last nympho instar to subimago. So in next, I measure the expression of the 4 key genes during metamorphosis in this species by choose from the genes that involved in metamorphosis: grouper homolog 1, E93, broad complex, and HR3. So to study the MECRE93 pathway in Creondipterum, we first measure the expression of 4 key genes that found in the whole body extract in the females of Creondipterum. For grouper homolog 1, we found that the expression start to decrease abruptly after molt to the last nympho instar, and they keep in the low level during day 1 to day 3. And then on day 4 of the last nympho instar, the expression of grouper homolog 1 increased rapidly, dramatically again, and keeps in high level in subimago and adult stage. In parallel, the expression of E93 is really low expressed during the penultimate and the last nympho instar on day 0, but then it starts to increase on day 1 to day 3 and show a dramatic increase on day 4 and keeps in the high level during subimago and adult stage. So for the expression pattern of these 2 genes, it suggests that the metamorphosis will take place during the transition from the last nympho instar to subimago, which is similar to the result that we studied the transformation of the morphology before. And the dramatic decrease of grouper homolog 1 expression on day 1 until day 3. It suggests that in this species, they also need the grouper-free or TS-free period, which is necessary for metamorphosis, like in the case of Bacteria germanica, that there is the decrease of juvenile hormone at the beginning of the last nympho instar. However, for the relation between grouper homolog 1 and E93 on day 4 or during subimago and adult stage, we still don't know what is the interaction between these 2 genes. Maybe it will have the interaction in some way, or the interaction of these 2 genes is happen and it may depend on the tissues dependent. Because we did another measurement on day 4, we separate each compartment of the nymph and measure the expression of Kruper or inhibitory and broad complex in the wings, in the epidermis, and ovary. And we found that for Kruper, the expression is more condensed in the ovaries or the reproductive tissue. But in the case of inhibitory, they show a high expression on compartment and broad complex. Even the expression during this period is already low, but we still see that is more condensed in the epidermis. So for the expression pattern of broad complex, it suggests that it should be involved with the wing development, similar to another hemimetabolant insect. And finally, a peak of HR3 that we found on day 3 of the last nympho insta and in subimago is suggests that there are two ecdysteroid pulse that trigger molting, first in to subimago and then to become adult. And finally, we did the experiment because we want to know how is the role of juvenile hormone in metamorphosis in this species. So we treat the nymph newly excised in the last nympho insta with the JH mimic metoprene. And the result showed that the juvenile hormone inhibits metamorphosis in this species. As you can see, they can live until the last moment of the last nympho insta when they have the black wing patch. But this metoprene treated nymph still produce the new mandibles, and they still have they still form the central filament, and the shape of the last tarsomere did not change to be short like a subimago. So this is the reason why I study the morphological change during the last nympho insta. So from this results, it seems like this metoprene treated nymph is going to morph to the supernumerary nymph more than more to subimago. However, when we measure the expression of the genes that involve in metamorphosis, we found the increase of Kruper homolog 1 when we treat with metoprene, but the expression of E93 and broad complex did not did not have any effect with metoprene. So this is the last experiment that I'm doing now. I'm treating with metoprene, but this time we separate each compartment of the nymph just to see, maybe we will see the effect of juvenile hormone on E93 in some tissues, like in epidermis. Yeah. And for summarize the results of this study, first the morphological study and the molecular analysis, and also the functional experimental in hormone treatment, it suggests that the metamorphosis in Cleon deuterum is take place at the transition from the last nympho insta to subimago, because everything has changed, especially during the last 2 days of the last nympho insta. From the expression pattern of the two key genes, Kruper homolog 1 and E93, during the first day of the last nympho insta until day 3, it suggests that the decrease of juvenile hormone is also necessary to initiate metamorphosis in this species, and is compatible with the McRae 93 pathway, because you will see the decrease of grouper and the increase of E93 during that period. And the administration of juvenile hormone mimic metoprene at the beginning of the last nympho instar. It can upregulate the expression of grouper homolog 1, but and metoprene can inhibit the formation of subimago. And from the result overall, we can say that the subimago would be the first phase of the adult stage, since we found that the metamorphosis is occur at the transition from the last nympho instar to subimago. So it should be not the pupal stage in homologous with the holometabolant insect. Yeah. And yes. So next, if the committee thing is appropriate, I would read the conclusions. Okay, thank you. So for the conclusions, the first 6 is refer to Blatella germanica, and the last 3 is referred to the mayfly Cleondipterum, the Paleoptera. So first, in Blatella germanica, myoclonine or male depletion in the penultimate nympho instar triggers the formation of the supernumerary nymph after the last nympho instar. Molecular analysis indicates that the expression of juvenile hormone acid methyltransferase JHAMT in the corpora allata became upregulated, which led to the increase of grouper homolog 1 expression in the last nympho instar and a downregulation of E93. From this result, we can conclude that myoclonine triggers the pre-metamorphosis stage by reducing juvenile hormone acid methyltransferase and thus juvenile hormone production. The molecular analysis confirms that the formation of supernumerary nymph is explained by the MECRE93 pathway. Second, myoclonine depletion in the penultimate nympho instar of Blatella germanica caused cell hyperproliferation in the protocyst gland, PG. Moreover, myoclonine depletion downregulates the expression of DACAPO, DAP, a gene coding for a cyclin-dependent kinase inhibitor involved in regulating cell cycle, and that of Neverland phantom and disembodied 3 steroidogenic genes. These results suggest that myoclonine induced the arrest of cell proliferation in the protocyst gland via the transforming growth factor beta signaling, which appears to be a prerequisite to stimulate the expression of steroidogenic genes and to produce a large ecdysteroid pulse needed to promote metamorphosis. Third, myoclonine depletion prevents the onset of protocyst gland degeneration. Our molecular studies have shown that the upregulation of juvenile hormone acid methyltransferase triggered by this depletion increased grouper homolog 1 expression and results in an upregulation of the expression of the inhibitor of apoptosis 1, IAP1, and a downregulation of the pro-apoptotic genes, Fujitarasu transcription factor 1, in the protocyst gland. Interestingly, the adult specifying genes E93 is also downregulated. These findings confirm the important role of juvenile hormone and inhibitor of apoptosis 1 in preventing protocyst gland degeneration, as well as the pro-apoptotic role of Fujitarasu transcription factor 1. Importantly, our findings also suggest that E93 is involved in the degeneration of the prothoracic gland in Blattella germanica. Fourth, depletion of E93 late in the final nymphoid insta triggers the formation of adult that preserve the prothoracic gland and can molt again in adulthood. These observations indicate that E93 plays a major role in eliciting the death of the prothoracic gland, which normally occurs after the imaginal molt in hemimetabolant insects and during metamorphosis in holometabolant insects. Fifth, previous studies in the laboratory show that the prothoracic gland death is induced by Fujitarasu transcription factor 1 in Blattella germanica. But our present results have shown that the Fujitarasu transcription factor 1 depletion that regulates the expression of E93, specifically in the prothoracic gland. So the above observations strongly suggest that the classic pro-apoptotic role of Fujitarasu transcription factor 1 is mediated by E93. Six, the treatments in the adult of Blattella germanica with physiological or pharmacological doses of 20-hydroxyecdazone 20E do not induce a new molt in Blattella germanica. These findings suggest that the adult epidermal cells are subjected to a source of adult commitment and do not respond to 20-hydroxyecdazone, apparently because they do not transduce the hormone signal. 7. The expression pattern of Kruper homolog 1 and that of E93 intersects in the last nymphoid insta of Cleondipterum prior to the subimago. This suggests that the metamorphosis in the mayflies take place at the transition from the last nymphoid insta to the subimago, and that it is regulated by MECRE93 pathway. 8. Administration of juvenile hormone mimic metoprine in the last nymphoid insta of Cleondipterum inhibits metamorphosis, so the treatment upregulated the expression of Kruper homolog 1, but did not apparently affect the expression of E93 and broad complex. These results indicate that Kruper homolog 1 inhibits metamorphosis, as in Neopteran insects. However, the observations do not allow to conclude that Kruper homolog 1 replaces E93 expression according to the MECRE93 pathway. Further work studying the expression of Kruper homolog 1 and E93, specifically in the epidermis, epidermal and reproductive tissues, should be needed to assess whether the MECRE93 pathway operates in mayflies. 9. The expression pattern of Kruper homolog 1 and E93 during the last nymphoid instars and the subimago indicate that the last nymphoid insta of Cleondipterum is homologous with the last nymphoid insta with non-polyopteran hemimetabolant insects, Polyneoptera and Planioptera. This leads to the conclude that the subimago would be the first phase of adult stage. And, yep. Thank you very much. Okay. That's all.